So today's program is sponsored by the Peterborough Town Library. Different libraries are sponsoring different speakers, and Peterborough Town Library is lucky to have Dr. Logan Brenner be the one they're sponsoring. So Mary, from the library, would you like to tell us a little bit about the Peterborough Town Library?
Yeah, happily. And thank you. I'm the assistant director at the Peterborough Town Library. And first, we're honored to participate with the Harris Center and our fellow librarians in this incredible opportunity to support Women's History Month through learning about the women scientists in New England and the work that they're doing. Living in rural New Hampshire as we do, we have innumerable opportunities to explore and learn. The Harris Center provides amazing resources for the community, as Susie just mentioned. And as I'm learning through these talks, so do our individual libraries, and I'm really inspired. At our library, Jocelyn Duffy, who has both experience in information science and natural resource management, has created a variety of nature-related programs. This winter, she organized a winter walk bingo card that encouraged patrons of all ages to get out into nature and see how much is really going on during this quiet time of the year. She also runs our short reads program, which meets monthly and discusses topics drawn from science. This month, actually on April 1st, the conversation will be about renewable energy and the Peterborough Energy Action Committee warrant article on the same. If you're interested, you can find information about that on our website. And in other news, if you live in the Monadnock region, you may have heard that we have a building project going on, just a small one. Anyway, I just wanted to let you know we are on budget and on time and hope to be back into our new old building by the end of August. So thanks again to the Harris Center. We're excited to sponsor Dr. Brenner, and I'm looking forward to learning all about the coral reefs and the history that they encompass. So thank you very much.
Thanks, Mary. Thanks for such a sweet shout out about the Harris Center. And we do love Jocelyn Duffy too. She's done some work for the Harris Center. We're big fans of hers. So keep up the great work, Jocelyn and Peterborough Town Library. It is really my honor today to introduce Dr. Logan Brenner to everybody. So today we're going to hear from Dr. Brenner. And I want to just say, if you have any questions as she speaks, you can put it right into the chat and I'll be managing the chat. And at the end, I'll raise those questions with Dr. Brenner. But let me tell you a little bit about her. She is a member of Barnard's Environmental Science faculty since 2019. She is a paleoclimatologist, and she's interested in reconstructing the past oceanic conditions using geochemistry of stony corals. So cool. A graduate from Skidmore College, she studied stalagmites as a way to develop the history of precipitation in the Yucatan of Mexico. She received her PhD in Earth and Environmental Science from Columbia University at the Lamont Doherty Earth Observatory, where she studied coral geochemistry, which I didn't even know you could study. So cool. Logan's research interests currently lie at the intersection of climate and environmental science. Her current projects involve studying the geochemical composition of coral skeletons to develop histories of ocean temperature, precipitation, salinity, and coastal factors such as river discharge. In addition to her research, Logan is passionate about science communication, science and policy, and the role STEM plays in higher education. So, Dr. Brenner, we're so happy to have you here. Welcome.
Thank you so much for that wonderful, wonderful introduction. Thank you, Harris Center and Peterborough Town Library, for having me. And it's truly an honor to be able to speak during this seminar series. And also, I love there's like a lot of Barnard love that I saw filtering through the chat. So that's wonderful to be in such good company. I have my virtual background is part of the Barnard campus. And quick apologies in advance. I'm coming out of my second vaccine haze. So if I get a little loopy, that's why. So bear with me. But yeah, today I'm going to be talking to you about how I use corals to study past climate change. So as you heard, I'm a paleoclimatologist, and you probably figured out from that description what that means, but we'll walk through it together. So what is paleoclimate? We can break the word down into two parts. It's essentially the study of past or ancient climate, right? So paleo always means like old, past, ancient, and climate is just the general weather conditions within a region over an extended period of time. You know, if you've heard of paleontologists, it's someone who studies old bones, old fossils. So that is what it means to be a paleoclimatologist. I may use this term interchangeably with paleoceanography, which is the study of past oceanography or past ocean conditions. And that's because all of my work involves studying the ocean. And although this is a New England-based group, we will be doing some COVID-safe traveling over the course of this talk to explore these corals. So why is it important to study paleoclimatology? Something that happened long ago. We have all these wonderful satellites. Why do we need to know about the past? Well, there are a few reasons why paleoclimate's important. One is that humans are just naturally curious, right? We want to learn about what happened in the past. We want to understand the place that we live in. It just helps contextualize our everyday experiences. Studying paleoclimate also provides baseline information. And what I mean by that is it helps us understand how climate varied before humans got here and started changing things around. So this allows us to understand natural variability in Earth's climate system, and then that can help us improve our predictions because we'll be able to tease out what I will be calling like natural factors versus ones that humans have a significant role in. And also we can just think about paleoclimate being important for the same reason that, we study history in school. It helps us understand who we are and the Earth that we live in, and perhaps make good decisions for tomorrow. So I want to start off with this. Now it's a slightly outdated graphic that was produced by Bloomberg, and it is going to show you changes in temperature compared to an 1880 average from 1880 to 2015. And you'll see as you get up on this side of the graph, we're reaching 2 degrees warmer than that average. Below on this side, it goes down to 2 degrees colder than that average. And this dark gray line represents observed or measured temperatures. So these are all temperatures that were either measured with a thermometer or a satellite, some type of instrument. So I will play this now. And what you're going to see are a few of the major natural factors that impact temperature, and they're going to start to fly by as different lines. So this is the impact of orbital changes or the geometry of Earth's orbit on temperature. This is how solar variability impacts Earth's temperature. And then we are going to see volcanic activity, which often leads to cooling of Earth's temperature. And we can add them all up and compare that to the observed record that we see. And it doesn't really approximate the temperature signal that we've measured. So now looking at some human factors, we have changes in land use, which impact temperature. We're also going to see changes in the ozone composition of our atmosphere impacting temperature. Aerosols in the atmosphere, which have a cooling effect, as you can see. And then the notorious greenhouse gases and how they warm the planet. We'll add up all of those human factors, and we can see how we approximate the observed temperature. So we're adding them up. Not bad, pretty good. But we have to include also the natural factors as well. And so that's what will happen next, the human and the natural. And we get an even better approximation of the measured temperature change. And so what's the significance of looking at this figure aside from these like lovely little lines flying by? So we can see that in order to understand temperature, we need to include both human and natural factors. And this also helps us figure out the role that paleoclimate can actually play. I said these are measured temperatures. I don't know how much I will rely on a temperature measurement from the early 1800s. I don't know how accurate those thermometers were. Even, satellites in their early stage were not super accurate. So paleoclimatology can help provide some more information or ground truth some of these measurements. And also, this record just goes back to 1880, which unlike our human experience time scale is pretty old. But if we think about, the 6 billion year history of the Earth, this is like nothing. And so how do we extend this record farther back in time? How do we understand, is this increase in temperature something to actually be concerned about? Is it something that's actually new? And that is the role that paleoclimate can play. Paleoclimate can help us understand how unusual is the climate change that we're seeing today and the climate change that's predicted for tomorrow and the future. So this slide has a few different images of different tools that paleoclimatologists use to study the climate of the past. The background image is the core repository at the Lamont Doherty Earth Observatory, which is in Palisades, New York. It's one of the largest core repositories in North America. And most of these cores are sediment cores that were taken from the ocean. So you have to take a big drilling ship out, and this is someone who is launching some of the drilling equipment. Those records can get up to millions of years old. They're quite special, and there are thousands of them at Lamont. And any scientist can fill out an application to use them. So it's quite a special place. You may have heard of the study of tree rings to learn about past climate change. You can see here these concentric circles. A dark and light band usually represents about a year of growth. And you can study the composition of the wood to learn about climate. This is an example of using corals to study climate. I'll obviously get into more detail about that, but just quickly want to also talk about ice cores, which is probably another method you've heard of in the news as a way to study past climate change. Typically, ice cores are taken from Antarctica or Greenland, and they go back thousands and thousands, hundreds of thousands of years, with the idea that ice from the top is younger and ice on the bottom is older. And so you kind of have this clock throughout the ice. And so I'm going to show you an example of how we can use ice cores to learn about past climate change. So there are lots of things that you can do with ice cores. They're a pretty powerful tool, but I'm going to explain how you can use ice cores to learn about the atmosphere of the past. So I'm sure you've all had to make ice for one reason or another in your freezer, and you take it out and maybe there are some little air bubbles trapped in the ice. And so if you were to measure the content of those air bubbles, it would tell you about what was in your freezer air. And so if you made ice tomorrow and then you checked that ice a year from now, you would be able to study paleoclimate and measure the composition of your freezer air from the year before. And so that's one of the ways that we use ice cores to study past climate change. As ice is deposited, these are examples mostly from Antarctica, but as ice is deposited, and there are little sips of air from the atmosphere that are trapped within the core. And then scientists can extract these cores. This is one way of extracting the core, measure the composition of these the tiny bubbles within the ice. And then you have this wonderful little measurement of the atmosphere from thousands of years ago. And there's this probably apocryphal story. I'm not entirely sure, of people who were studying ice cores, and it was July 4th, and they took some ice from 1776 and cheersed with it. And I don't know how accurate that is, but I think it's kind of cute and funny. But anyway, back to the actual science. So what we can see here is some measurements that were made by studying the composition of the little bubbles within the ice from a core that, again, in terms of paleo timescales, it's not so old. It goes from 2000 CE back to 1000 CE. But what we can, the scientists measured methane and carbon dioxide, two really powerful, important greenhouse gases, over time. And what they found was that things were pretty consistent until about the 1800s. And this is pretty much where the time period of that animation that I showed you. So this helps contextualize and answer that question: how unusual was that climate change? And scientists, we have also established a relationship between CO2 and temperature, so we can make that jump. And so this is just one way that that we can use the natural world to learn about past climate and contextualize that change. And it's a pretty shocking level of stability compared to this huge increase in greenhouse gases in more recent times. But I'm not here to talk to you about ice cores. I'm here to talk to you about coral reefs, which are my tools for studying past climate change. And I think when most people think about corals and coral reefs, they probably think about biology and conservation, which are incredibly important. That's not quite my area of expertise. I actually mostly view corals as tools to study past climate change. Although I am very concerned about their conservation and preservation going forward, I'm most interested in how I can use them to learn about the past rather than just learn about their growth in general. So these are just some images from Turks and Caicos, which is where I spent my time studying abroad in college, which was a great experience and my first experience doing like oceanography research or marine science research. So this was me a few years ago. I don't know, I think I was counting corals at the time. But yes, this is the coral reef. And for the purposes of this talk, when I refer to corals, I'm talking about scleractinia, which are stony corals. Corals is actually quite a broad term, but I'm not talking about these like sea fans here, you know, the ones that like sway in the waves. That's not what I use in my research. I'm more interested in these large corals. And I'll show you a few more images later of the types of corals that I end up analyzing. But just to make sure everyone's on the page, I want to go over what is a coral. And so I'm going to start off with this slightly inaccurate definition, but I think is interesting, or slightly inaccurate equation, but I think gets the point across. So corals are living rocks. That is completely true, but this equation is a little bit inaccurate, right? So corals are these large rocks, but they are alive. They are actually carnivorous and are able to photosynthesize, hence the inclusion of a little Venus flytrap over here. So although they don't move around, they are animals and they do a lot of really crazy, interesting things. So they are living carnivorous rocks. And in reality, in the oceans, this is what they look like. Sometimes they're plating, it looks like they have fingers. This is kind of like a brain coral. And this is a really large domal coral from the Pacific Ocean and Fiji, and it's of the genus Porites. And these are like the money corals that we use in paleoclimate. They're huge. They provide a long continuous record of their surroundings. So this is really the types of coral that I'm interested in. And I just want to go a little bit deeper into how corals grow so you can understand a little bit more about them. This is a many times magnified schematic. This is the coral skeleton, and this is the coral animal. This is actually only a few millimeters thick. The living animal is quite small. It's just a thin coating around that whole domal structure or whatever structure. These tentacles have little tiny stinging cells in them. It wouldn't hurt you if you brushed against them unless you brushed against a fire coral, as the name suggests. Stay away from those. But this is the tentacles that are used to capture prey, and then they bring the little prey. It's like tiny photosynthetic or little organisms, sometimes microscopic, into their mouth. So that's the carnivorous aspect of the coral. This is called a polyp, and this is the animal. It's quite simple. There are no eyes or anything. It's a very basic animal. Within the, but I also said they're photosynthetic. So within the tissue of the coral, we can magnify it here, there are these little zooxanthellae, which are tiny photosynthetic algae. And a coral is like a perfect example of a mutualistic symbiotic relationship. So that means the coral animal and the tiny zooxanthellae benefit from living together. And so the zooxanthellae photosynthesize within the like safe home of the coral tissue, and then the coral benefits from the byproducts of photosynthesis. And although they're carnivorous, they actually meet upwards of 90% of their energy needs via photosynthesis. So that's really quite important. And the coral all the time as it's growing is secreting its own skeleton, the way that we secrete our own skeleton. And that's made up of calcium carbonate. The specific mineral structure, the way that that mineral looks, is the form of aragonite, but that's not super important for the purposes of this talk. But this is what I am most interested in, is this skeleton, this calcium carbonate. And again, calcium carbonates, it's what's in Tums, it's what's in chalk, or eggshells. So it's garden variety calcium carbonate. There's nothing like too special about it. You've interacted with it multiple times. And so this is what I'm interested in. But before I go just more into the proxy, I think I'd be remiss if I just didn't quickly talk about bleaching, as it's something that I'm sure you've heard about before. Coral bleaching is what happens when the coral gets stressed out for whatever reason. They exist and thrive within a very narrow range of environmental conditions. And when it's perturbed for an extended period of time, they end up expelling their zooxanthellae. So these little photosynthetic friends that they rely on for most of their energy needs. The reason, and if they're not recolonized by zooxanthellae, then they will eventually die. It's not necessarily a death sentence as long as they are recolonized by zooxanthellae. So why is it called bleaching? Well, zooxanthellae are responsible for the color of corals pretty much. And so when those zooxanthellae are gone, all you see is this white calcium carbonate. So that's why it's called bleaching, because they go from having these wonderful different colors to being stark white and looking like the chalk, that we are used to when we think about calcium carbonate. So now that you know about what a coral is and a little bit about how they grow, we can talk about how we use them for the study of paleoclimate. And so corals provide what's called a climate proxy. And a climate proxy is essentially a method of providing information that stands in or indirectly represents climate conditions. The same way that if you couldn't go to a committee meeting, you would send a proxy on your behalf. That's the same term and same way we're thinking about climate proxy here. So corals are an indirect representation of climate. And we can dig a little bit deeper and look at and get into the geochemistry aspect, but I'll just quickly have another little schematic to explain this transitive process from climate to coral. So we can think about, there's some aspect of climate that we're interested in, temperature, precipitation, salinity, you name it. These are just the two little clip arts that I chose, but there's some, change in climate. And then that change in climate impacts the chemical composition of seawater. A simple way to think about it is, if it was very rainy in a given area, the ocean underneath it would probably be a little fresher because of the influx of precipitation. So climate changes impacts the composition of seawater. Then that composition of seawater is incorporated into the coral skeleton. And for this, you can think about the phrase, you are what you eat. The coral can only be made up of what is in the ocean around it. And so it's that calcium carbonate skeleton reflects the composition of the seawater. And there are subtle changes that happen to the skeleton that reflect the chemical composition. Maybe it's the isotopes of oxygen that are included, or there are some metals that look like calcium and can substitute in. And so there's subtle changes to the skeleton that are then recorded within the coral over time. And then eventually we can measure the coral skeleton through our various lab methods that I'll talk about, and learn about the climate. And this is an X-ray positive of a coral. So a core that has been taken out, and then just, if you've ever had a regular X-ray, the same exact machine that we use here. And you can see those bands, which are reminiscent of what you saw in the tree rings, so that they do function similarly. So if we want to go from coral to climate, we just have to follow this in reverse. We measure the composition of the coral skeleton. That tells us something about the composition of the seawater. And then we have to have some pre-established relationship that then gets us from composition of seawater back to climate. So this is the basic premise for all of the coral proxy work to learn about the oceans. So there are tons of different coral proxies out there, or rather there are tons of different aspects of climate that we can learn about based on the geochemistry of corals. So I've listed a few of them here. Sometimes we're just curious about what the seawater was made of. Sometimes we want to know about past temperature, salinity, precipitation, flow of rivers, land use changes, pollution, even wind strength, right? So wind can carry different amounts of dust, and nutrients and organic material. So there are tons, and this is not an all-inclusive list. There are tons of aspects of climate and environment that corals can tell us about. Corals are also really special because they provide different durations, different durations of records. So you might have a large coral that provides like a continuous record that's hundreds of years old. Or you might have found a smaller fossil coral that obviously wasn't still living when you found it, but provides a little snapshot from thousands of years ago. So they're quite flexible in terms of like the duration that you want to study. The other thing that they provide is the potential for really high resolution records. So corals, depending on the species, can grow maybe 10 to 14 millimeters per year. And so that means if we sample our corals at, a 1 millimeter interval, we can almost get monthly, monthly resolution. And that's pretty powerful to be able to go back hundreds of years and get sub-annual variability in our records. Those ice cores that I showed you, that's great. They go back thousands of years. They're not going to tell you with this specificity of resolution. Those sediment cores I told you that go back millions of years, that's great, but you're going to be looking at like time slices of thousands to millions of years. So corals really give you that high resolution opportunity. Of course, you can also look at something on an annual cycle by averaging or maybe sampling at a different rate. Or maybe, and what's more common in fossil corals is you might just get a mean state. So averaging all of the material from your little coral. And that's still quite valuable when you're thinking about wanting to understand climate. Maybe if you have a coral that's 10,000 years old, it's still great to have just some information, even if it's not, this is what happened in January 3,000 years ago. So a little bit about the coral collection process. I do feel like I need to disclose I'm not in any of these pictures. Unfortunately, I haven't had a chance to drill out corals for this work. But nonetheless, I want to explain and show you how it works. So first we need to collect corals. These are some of my collaborators who were diving in Fiji. They found this wonderful coral that provides this long continuous record. And cylindrical cores are then removed with a hydraulic drill. And it's really important to do as little damage as possible to corals. Obviously, this is a destructive process. And so what we often do is patch the holes up with cement, and then tissue layer will regrow over the hole. The cement plug also prevents boring organisms from entering that little hole and then like starting to eat at the coral, which could impact the structural integrity. That's just a very small way of potential of like reducing our impact. Now all scientists have to publicly store their data. So the idea is that not only is it accessible to everyone and makes science a little bit more equitable, but it could also reduce the need to do field work. And so personally, I try to use our corals and milk as much out of them as I can. To avoid having to make a big impact, because if you were recreationally diving, you should never be touching a coral because this, they have knees on it. It's not good practice, and it's quite difficult often to get permits to conduct this work because everyone is trying to protect their coral reefs. They are quite fragile and ecosystems that are at risk all of the time these days with ocean temperatures on the rise. But nonetheless, they are quite powerful proxies. And so we do try and sample them with as minimal damage as possible and get the most out of them that we can. So once we get the corals into the lab, this is where most of the time is spent, which is in sample preparation. So these are the cylindrical cores that you can imagine were extracted from those large coral heads. And the next step is using just like a water-cooled tile saw to take out like a flat slab from the middle. So it'd be pretty hard to work with something that's round. Then we can X-ray them. I'll show you some more X-rays in a little bit. And we can sample them. So if we want to go back in time, we need to drill down what we call the maximum growth axis. This is just actually like the paper ruler that you get from IKEA. Those work really well. And so we tape them along the corals so we can keep track of the way that we've been sampling them. This is at millimeter scale. So these from like here to here is 10 millimeters, just to orient you. So this ultimately gets up to 100 millimeters. And there are two main ways that, at least the way that we drill, we sample our corals in our lab. I say our because I have my lab at Barnard and I work very closely with people at the Lamont Doherty Earth Observatory. So one of the ways is using this mounted mill, and it's just kind of like hand cranked. It moves forward maybe 0.5 millimeter. You collect the powder, you store it. So you end up having this like all of these coral powder samples that go back in time. The other way that I use more frequently is I hand drill the corals. And I use a Dremel drill. Maybe you've used it for home improvement projects or arts and crafts. And if you've had a cavity, then you've probably experienced the type of drill bit that I use in the drill. So it's I go to like a dental supply website and get some like diamond tip little spherical drill bits, put it on my drill from Home Depot, and then I drill out at the 1 millimeter scale. And so you can imagine if I have like a 3 meter long core, it's going to take a really long time to drill out at 1 millimeter scale. So it's quite tedious, but what you need to keep reminding yourself is, okay, I'm going to have this record that's really amazing. It's going to be high resolution, go back hundreds of years. So you need to like keep telling yourself that, so you don't, go cross-eyed when you're doing this in the microscope. So that's the way we drill out powder. We store it in vials. And then we want to, so that's like archived powder. And then we can take out a little bit when we want to conduct analyses, the geochemical analyses. So when we want to know what type of metals have been incorporated into the skeleton, what's the isotopic composition. And so this image shows you about 250 micrograms of coral powder. This spatula is maybe like an inch long. So you really like have to kind of hone your fine motor skills within this balance. Static is not your friend when you're making these measurements, because the powder just kind of evaporates. So again, it's really like a study in patience. And so then we measure out whatever we need for analysis, we archive the rest of the powder, and then we can move on to our next steps. So I'm not going to explain how these machines work, but just want to show you the main tools that we use to conduct the geochemical analysis. So to get from calcium carbonate to then some type of like meaningful, then to get to the meaningful proxy information. So actually figuring out, what is this coral skeleton made of? So on the left here, we have an isotope ratio mass spectrometer. So we use those often to measure the ratio of different oxygen isotopes within that calcium carbonate skeleton. And that ratio of heavier isotopes to lighter isotopes is impacted by things like temperature of the water or the salinity of the water. The other machine that I commonly use is and this is more of a mouthful, inductively coupled plasma optical emission spectrophotometer, which is conveniently shortened to ICP-OES and even more conveniently shortened to OES. So if I refer to it, I will refer to it as the OES from now on. And this machine helps us conduct what's called trace metal analysis. Oftentimes we'll be measuring strontium or barium in the coral skeleton. So those are the two machines that I commonly use. And just to give you a little bit more visualization about some of the lab work that goes into these projects, I have a time-lapse video. It goes by very quick if you're like, it's not like quite a strobe effect, but if you're not into things that move very fast, look away, I'll tell you when it's done. But these are some of the steps for weighing, preparing our vials for various analysis, lots of preparation. Here I am preparing some dilution standards for the OES, which helps calibrate the machine so we know how to orient ourselves to the values that it spits out. And then the next step that we're going to show is diluting or digesting those calcium carbonate samples. So I'm just adding some nitric acid to these samples. That's because the machine, the OES, measures things in solution, not solids. And then mixing them up to make sure everything is homogenized. Putting together some of the tubes on the OES. And then this is the part where I can sit down and I watch this machine go for a while. And again, this machine can maybe analyze 60 to 90 samples in a day, depending on if it's, by the way, if you looked away, you can look back now, the video has stopped. But this machine analyzes maybe 60 to 90 samples a day, which is, and like that's depending on if it's working well and when I get into the lab. But that's, you know, maybe that's not a lot of years of data if I have 10 samples per year. So again, this is a really long, it ends up being an expensive process. But again, you just have to think about like the payoff at the end, which is that you get these nice, long, high resolution records. But, you know, sometimes you're limited by things like your grant money and you can't analyze every sample. So it's, you know, all a balancing act. So I now want to actually go into a case study. One of my projects in progress that involve coral paleo work. We'll see how much time we have. If there's time, I can show you another project, but I just wanted to focus on this one first. So in this case study, I will be talking to you about proxies for river discharge, which is you can think of it as river flow, river flux. The technical unit is like unit of volume of water per second, so it's a rate. So it's, you just think about it as a lot of how much water is moving through the river. And a bigger number means more faster. The record durations that I'll be working with are continual records that will span from decades to centuries. I'm only at the decade point at this point, but you know, post-COVID, I'm looking forward to analyzing more samples and getting up to centuries. And then I will be showing you these near monthly records, and then some other averages. So this is just one, you know, one example of how I'm using corals. And so this study is formally, you know, called the development of a multi-coral barium calcium river discharge study along the Pacific coast of Panama, another mouthful. But essentially, what I'm doing here is establishing a relationship between the amount of barium in a coral skeleton and river discharge to learn about hydroclimate, so all things water, along the Pacific coast of Panama. So why should we even study river discharge in Panama? It's always really important to make sure to make sure you understand why you're doing what you're doing, and to put your time, your money, your effort, your emotional energy into a project that makes sense, and not one that just exists because it exists. So why study river discharge in Panama? Why is it important? Well, there's really strong wet-dry seasonality. There's a very clear wet season and a very clear dry season in Panama. This has to do with some of the atmospheric cells above it. And so it's an area where there's great potential to learn about when climate's very wet and when climate's very dry and how that impacts coastal waters. Tons of rivers also empty into the Pacific coast of Panama. It wouldn't make sense to study river discharge if you were at a coastal region where there were no rivers, right? So you need to do something that makes sense. And what I have here are the locations of my two corals. They're located in the Gulf of Cherokee. That's about this area here. This coral is closer to shore at S1, and this is by a larger island, Coiba Island, and we call this coral IC4A2. We need some like, I don't know, some peppier nicknames, I think, for these coral IDs, but here we are. So, and there are tons of rivers that discharge into the Gulf all around here. So why are we studying river discharge and Panama specifically? Well, river discharge records in Panama and in general are not amazing. And by not amazing, I mean they're often short, they might be discontinuous, slightly unreliable. So like we could never do that animation that went back from, you know, 1880 to 2015 with river discharge measurements. We just don't have that information. And so this is where the coral can step in and say, I'm going to fill in those gaps. So that's a great reason to study river discharge. Eventually, once we have those records, we could identify anomalous events. By anomalous events, I mean like very dry seasons, or maybe even a dry season that's kind of wet. We might be able to understand the impacts of El Nino a little bit more, a very relevant climate phenomenon for this region. So once we get this baseline record, we can then kind of manipulate it a little bit to learn about different modes of variability. And, you know, if you're thinking about Panama, you have to also think about the Panama Canal, which is pretty critical to global trade. The canal is more over here. But what's really unique about the Panama Canal is that it's completely driven by changes in hydraulic head. So that means that it's totally pumpless. And so water moves through the canal as managers let water flow from like reservoirs into the canal or out of the canal into the reservoirs. So they're just moving water around. And so you can imagine if it's quite dry and there's not a lot of rain or rivers feeding these reservoirs, you're not going to be able to kind of increase the amount of water in the canal. And so what happens over and over again is they put restrictions on the size of the ships that can travel through. And so it's really helpful to understand, to maybe know in advance, oh, this is going to be a tough year because I know what happened in the past, and maybe be a little proactive about water management. So that's kind of like the long-term goals of understanding past river discharge here. So I also want to show you some of the available instrumental data. So in blue, we have rainfall, and in green, we have river discharge, and river discharge is shortened to the letter Q. And so there are a few things that we can see here. There's a clear peak and a trough pretty much every year in both rainfall and river discharge. And that's because of this really strong wet dry seasonality. And it's always wonderful that like our instrumental data reflects what we think is happening. It's always important to check. The other thing to note here is that river discharge and rainfall are largely aligned, which is not always the case. Sometimes there might be a lag in the system, or maybe groundwater is influencing rivers that is, not reflected in rainfall. So the main takeaways from this figure is that there's clear wet dry seasonality in both of these records, and it appears that river discharge is impacted by rainfall. The other thing is you can see that this river discharge record only goes to like 1971-ish. And I stopped the record here because this is when how old our corals get. So there's a lot more to learn, right? We only have like 10-ish years of data from the river discharge record. So I want to now explain with my super high-tech graphic how the barium calcium river discharge proxy works. So what we have here is our reef, along the coast, and it's right up against the land, which are these brown rectangles, which we have a river running through it. And all of these black dots are barium. And now in the ocean, there isn't really a strong natural source of barium. And that's because most of the barium comes from terrestrial materials like rocks. Barium is much more common on land than in the water. So what we're going to see here is we're going to have some river flow represented by this arrow. And when there's some river flow, barium erodes out of, or some of the terrestrial material erodes out of the land, gets carried by the river into the water, at which point, due to various chemical properties, the barium is then released from the physical matter, like from the little sands and grains that it was attached to, or that it composed. It enters the water and then can be incorporated into the coral skeleton. So erodes from the land, the rivers take it into the ocean, deposit in the ocean. Now the corals can incorporate it into their skeleton. And this is because calcium is an ion that is a 2+ ion, and so is barium. And so the coral doesn't really care that much. I'm anthropomorphizing it. It really doesn't have any feelings when it's making its skeleton, but essentially it gets tricked. It can incorporate barium in the same way that it incorporates calcium because it's the same size and it's the same charge. Okay, so that was what you saw what would happen when there was a little bit of river flow that brought barium. Now you can see I'm increasing the amount of river flow denoted by this larger arrow, and it's depositing a lot more barium into the coastal oceans, and then a lot more barium can then get incorporated into the coral. So it's a pretty straightforward, the basic principle is pretty straightforward, which is that more river flow or more river discharge will bring more barium into the coastal waters, and then more barium can be incorporated into the coral skeleton. So now to show you some more squiggly lines for my corals. Over here on the right is some of those coral x-rays. We can see those annual bands, those dark and light bands. These are x-ray positives. And so that means the darker material is denser And the darker material is skeletonized during the wet season. And that's because photosynthesis is lower, because it's cloudier, it's stormier. And so it's just not depositing as much material. In the dry season, photosynthesis increases, the coral is growing more rapidly. And so it's less dense material and it's whiter. And these are our two corals. This is coral this is coral IC4A2, which corresponds with the red line. And this is S1, which corresponds with the black line. And this red line here just shows where I drilled the sample, or where I drilled down the entire slab. And so this is just some of the preliminary data that we have. We have time and calendar years on the x-axis and the ratio of barium to calcium on the y-axis. This coral goes back to the early 1900s. This one goes back to 1707. So we have a lot more work to do. But what we can see here is that the two corals match up pretty well, which is always nice to see. They are living organisms, and sometimes they just don't end up calcifying the way we think they will, because other things are happening in biology that I will not pretend to understand. But things just sometimes happen. So it's really wonderful. We see these great annual cycles, similar to what we saw in river discharge and rainfall, which is a great sign. And the two corals appear to reflect one another. These are all things that, based on those like basic assumptions, are great, but we can't take them for granted just because there are always things that could happen in the system that we don't know about. And then as the values increase, it suggests that there's more river discharge. When barium calcium is lower, it suggests there's less river discharge. So then what I'm doing over here is I'm then taking some averages from the wet season values of our coral, which is the orange line now, and it's a composite coral, so it's just an average of these two together. So now there are only 2 points per year. There's the wet season value and the dry season value. And I've done the same treatment with the river discharge record. So what we see here is this excellent correlation at the seasonal level of river discharge and barium calcium. And so, like I said, it's really, we can't take this relationship for granted. There are tons of places where this proxy doesn't work for other reasons. There might be other factors that are just overprinting the geochemistry, or there's just an unknown reason that the proxy doesn't work. So this was really wonderful to see them so well correlated. And then the next step is figuring out how we can translate river discharge to barium calcium. And so what we did was apply a regression. This is no longer by time, but these points are still like time matched. And so we applied a regression through all of our data. It's not much data because we are limited by the amount, by like the 10 years of instrumental discharge. And again, what we see is a really wonderful fit between river discharge and barium calcium. And so this suggests that barium calcium is largely driven by river discharge. And now we have this statistical mathematical relationship that will allow us to convert river discharge to barium calcium, or eventually when we go back in time and we don't have instrumental data, we can convert coral barium calcium to river discharge. So that's like, those are units that we can understand a lot better than, micromoles per mole. Like that doesn't have much meaning for us. But if we can convert it to meters cubed per second, then that really helps us understand how river discharge changed over time. And so there's a lot more work to be done. This was a really wonderful first step, but now it's promising, and there's more to do and much more to learn. So some of the main findings from this study, as I have gone through, I've mentioned them throughout, but just to summarize here, barium calcium in coral skeletons is influenced by river discharge in the Gulf of Cherokee. Like I said, this isn't the case everywhere. And so it's important to always validate your proxies first with the youngest data that you have. And if you recall from that site map, those two corals are pretty far apart. They were about 74 kilometers apart. And so it's pretty striking how similar they are, which supports the use of adding more corals to this network. And maybe we can improve that statistical relationship even more. So that's another step, is just to analyze, once we finish analyzing these corals, bring some other corals into the mix, reduce error, improve the calibration, and have something that is maybe more representative of the whole Gulf. And eventually, with further development, We can use the calibration and these longer records to identify those atypical hydroclimate concerns, maybe an extended drought, maybe El Nino, something that has a lot of meaning for human practices every day. And so this was kind of the first step in this process, was evaluating this network approach, evaluating the proxy, and now we can go back in time and complete these coral records. And just to, more of my pro paleo coral, spiel is that I think I'm obviously biased because I work with corals all the time, but I think they're a wonderful climate proxy. But to summarize, they provide information on flexible time periods. I showed you an example with modern corals, but I also do work with fossil corals, which is really cool. They can fill in unknowns when and where instrumental data are not available or when it's unreliable. And you can also generally learn about the conditions of the coral reef. So although I said I'm not like a conservationist or a biologist who studies modern reefs, a lot of the byproducts of my work is relevant to those people. So like the next step is figuring out how to work with them, to improve, so we can improve both of our findings. And then of course, understanding what happened in the past provides us baseline information to help us tease out the modern and projected human impact and figure out if we can make better predictions for tomorrow. So these to me are just some of the main points of the power of paleo with corals. And with that, I would really like to thank you for having me and for your time. And we'll be happy to take any questions, and whatever I don't get to, you're welcome to email me and we can chat that way. But thank you very much.
Wow, Logan, thank you so much. That was really fascinating. And we do have a bunch of questions. I see people applauding, and there was lots of people kind of talking about great questions. So Miles actually had a question, and I thought, Miles, would you want to ask your question?
Sure. Do the management of the Panama Canal follow your work? Is there interest from governments? And the larger question of where does your funding come from for this kind of research?
That's a really great question. So part of, so some of this work is funded through NSF, the National Science Foundation. So that's through the US. We had said in that grant that we are going to share our work with the what is it called? It's like the Panama Canal Authority. So we do have one contact there. They're not like actively following each step. I don't think that it would be I don't think they would care that much about like the nitty gritty here, but we do intend to share with them. And I mean, on a more personal note, and even just like the past year, I've been learning a lot, trying to self-educate more about environmental justice and working with local organizations. And if I had to write that grant again, it would be a little bit different. And there would be a lot more local involvement because that's really important, that the data gets into the right hands and that there are local people who are involved who know more about some of these conditions than I do because that's where they live. So going forward, definitely would like to do more interaction with, policymakers and local organizations, but there is some that's planned.
Great, thank you. That was really interesting. Yes, so there's another question here about how is it different sampling corals in freshwater lakes like Lake Champlain that was once a formerly inland sea? Is that anything that you do, and is it a different process?
I'm not super familiar with if there are corals in Lake Champlain. I imagine if you are asking if that there might be, there needs to be like some salinity, it can't be, I guess, totally fresh. There are a few differences. There's also like deep water corals. So the process would be similar in that you could study the geochemical composition of the coral, and that will reflect the conditions in which it grew. But the way that you actually physically sample them is probably different because they look different. And they grow slightly differently. Like I said, I'm not familiar with what's in Lake Champlain, but there are a lot of deep water corals that are called bamboo corals, and they grow more in these like concentric circles. So for those, you're kind of like drilling in towards the middle rather than drilling down a transect. So that's one difference. So yeah, it would be mostly the sampling protocols would be different. I don't know how long-lived they are either.
Cool. Okay. Here's a question I wondered about too. It's from Norma. She's wondering if there are hydro dams or agricultural dams on the rivers that discharge into the Panama Canal, and can you tell that from looking at the coral?
Yeah, dams are a big pain for us because when we're looking at when we're trying to create that like regression with that modern data, sometimes the river discharge values are impacted by whether or not the rivers were dammed. And that information isn't really easy to come by. So that's really a limitation. We need to do more work into where these dams actually are. But I would say it becomes more of an issue rather than discharging into the Gulf directly, but impacting the instrumental data that we have to establish that calibration. I think that's where it becomes more of an issue.
Cool. All right, here's a question from Jeremy Wilson, our executive director of the Harris Center, and his background is in forestry. And I think he's thinking about trees. He's wondering if, do the year lines in the coral x-rays not show up in an area without a strong wet season or strong wet season or dry season?
So typically you will find okay, step back. They also show up when there's maybe like a very strong like summer and like winter. So maybe like a really strong temperature change. So essentially when there's just a big difference throughout the year in one aspect of climate that could impact calcification. So in general, there's going to be some level of banding, but we don't strictly rely on the banding to create our chronologies. We also do some wiggle matching of like that, you saw the barium calcium record. And then instead of having it over time, we'd have it by like millimeters, and we'd kind of match it up a little bit to some of the instrumental data that does have time.
Cool. Are corals the only kind of things that can give you such a month by month or almost month by month information back through history? Or are there other things that you know of that also could provide that?
There are some other ones. I think corals are the best. You can get some good resolution with tree rings that can be subannual. And again, I'm probably overstepping when I say monthly, because we don't know exactly is this January, but we can say, okay, we have about 10 to 12 points per year. So you can certainly get subannual resolution with other proxies, tree rings, maybe some other terrestrial materials, but it's much rarer to find.
I think that might have been that. I think we're just about out of time. That might be our last question. If you didn't get your question answered, you can reach out to Dr. Brenner at Barnard. And wow, I just want to thank you. There's lots of people thanking you in the chat saying it was great. They loved your slides. It was really informative, really fascinating. So thank you so much, Dr. Brenner. And we hope we see you again sometime in the future doing another Harris Center program. It was wonderful.
Yes, this was wonderful. Thank you so much for the invitation. It was great.